Good morning, everyone, and welcome to Lunch with a Scientist. I'm Leah from Headwaters Science Institute. Today we're going to meet another great scientist who's a PhD student at the University of Nevada in Reno. But before we get into that, I've got a couple reminders for you. If you've been watching the series, of course, you're probably familiar with this already. But the first thing we have coming up is this awesome talk on March 5th with Robert Swan. He's a world traveler, he's a climate activist, and he also happens to be training for his next expedition here in the Tahoe area where we are based, down at Royal Gorge, towing a little boat around, trying to get back into great physical shape for his next expedition through Antarctica. So we're hosting him on March 4th at 5:30 in the evening. Please join us. This talk is free and sponsored by Headwaters, and we also want to thank Tahoe Mountain Sports for sponsoring the talk as well. You're welcome to donate if you'd like, but otherwise tickets are free, and this is totally suitable for the whole family. If you can't make it live on March 4th, you can also watch the recording. It'll be up for 48 hours on that same YouTube link that you can get through registering. So please join us. We'd love to see you there in the virtual theater. And the other thing that I want to share is that we are announcing our upcoming summer program dates. So this summer, we've got our girls' summer research experience, and we also have our regular summer research experience for both boys and girls. You can join us through this virtual program online where you'll learn how to create a research project around anything you find interesting, author a research paper, and work directly with a professional scientist mentor. So it's a really good opportunity to get an experience for your college application. And then, of course, in the girls' camp, you can work specifically with a female mentor and be inspired by what other girls in science are looking to do. So please feel free to join us for those camps. This is our little teaser of our camp dates, and registration will be going live in about 2 weeks, so make sure you check back on our website on our student program page. So with all of that said, I'm really pleased and excited to introduce Keely Rodriguez today. She's a PhD student at the University of Nevada, Reno in the Ecology, Evolution, and Conservation Biology department. She has worked with many endangered species, including fish, sea turtles, and frogs, and she currently studies frogs, looking at their immune systems to see how they react to deadly diseases. So she's going to tell us more about that today. And she says that once she obtains her doctorate degree, she plans to pursue a career of teaching and research. So we're really happy to have her step into that teaching role today and share with you some information about how amphibians deal with disease. So with that said, I'm really excited to welcome Keely Rodriguez. Hi.
Hello.
How are you?
How are you?
I'm good. How are you?
Doing very well, thank you.
Great. So I know that we're going to jump into your talk really soon and hear all about frogs, but there's a burning question on my mind. It's winter right now. For most of us, there's snow on the ground unless you live in the tropics. And what exactly do frogs do in the winter?
That's a really good question. So yeah, in the tropics, they don't have big bipolar seasons, so they just kind of hang out year round. But in the temperate areas, such as the desert and further north and south of the equator, the frogs will employ a lot of different techniques, but some of them involve burrowing into mud or sand or using another animal's burrow to kind of go into like a hibernation or brumation state. And in really cold areas where it freezes, the frogs will actually, and amphibians in general, will actually store minerals and molecules have a higher percentage in their body, such as sugars, and that actually prevents the frog from freezing in a freezing condition. So the outside, like maybe fingers or toes, might have a little bit of frost on them, but the internal organs stay warm and healthy and the whole time.
Fascinating. So as we're watching along, we'll know that frogs are still out there, even though it's chilly out.
Yep, they'll be back.
Great. Well, Keely, we're really excited to hear about your research. So I'm going to pull in your presentation, and I'll let you take it away.
Awesome. Thank you so much. So I'm really, really excited to share my research with you all. My research is entitled Saving Frogs with Science. And today I'm going to talk about my background and work. Then I'll tell you about amphibians and chytrid. And then at the end, we're going to search for some gold. You're going to come with me on a hunt. So quickly, some background on myself. I grew up in Las Vegas, just 7 hours of here, south of here. I went to Nevada State College, and I got a bachelor's degree in biology. And then I started UNR in the Ecology, Evolution, and Conservation Biology department, where I study disease ecology. So disease ecology is defined as the study of diseases of species and how these disease dynamics shape ecological processes. And we'll come back to that a little bit later. Disease ecologists work outside in the field collecting data as well as inside the laboratory performing experiments. I also have the coolest job ever. My research requires me to problem solve. It can be extremely challenging, like a puzzle, but I get to do laboratory experiments that no one has ever done and use all kinds of technology to find the answers that I want. So let me introduce you to some of our lab equipment. This machine here is called a mass spectrometer. If I want to identify an unknown molecule, I will put it in this machine and it'll blast it with electricity, and the vapors are analyzed by a computer, which tells me what it is. This machine is called a thermal cycler. I use this machine to detect DNA, and it's exactly the same machine that researchers studying COVID use to find out if you are infected if you get tested. And let's see. And then this machine is called a spectrophotometer. So this machine can tell you how much of something is in a sample by shooting light into the sample and then measuring how the light changes as it travels through the sample. But it's not all easy. Sometimes you get your car stuck in the mud. Sometimes your study organism jumps out of your hand down into a hole, and sometimes you come face to face with some of the damage that humans can do. But sometimes you come home from a long, tiring night shift to see this sunrise. Sometimes you watch endangered baby sea turtles make their trek down the beach to the ocean after you have weighed and measured them. And sometimes, if you're very lucky, you may find a species that was thought to be gone forever. So disease ecology is very similar to the job other researchers do with COVID-19. They work to find out how the disease infects humans and spreads, how humans can prevent and fight off infection, and then how the pandemic has affected the whole world. So now that I've given you a bit of background on what I do, now I can discuss why I do it. Amphibians and chytrid. You see, decades ago, researchers would stumble upon scenes such as this. Masses of dead frogs, or even more sinister, no frogs at all. They had been killed by a pathogenic fungus that infected their skin, called Batrachochytrium dendrobatitis. There's lots of different ways to pronounce that, but I'm just going to refer to it as chytrid to make things easier. Chytrid was discovered in 1998 after researchers worldwide were perplexed about why the amphibians they were studying were suddenly dying. And since then, chytrid has caused numerous population declines and also some extinctions. You see, the important thing about frog skin is that along with lungs, frogs can use their skin to breathe. So here we have a picture. This is the epidermis, this is the frog skin, and then you have a highly vascularized area at the surface of the epidermis, which can allow for gas exchange, such as oxygen and CO2. Similarly, they use their skin to obtain important other electrolytes like sodium, potassium, and calcium, which play a really important role in muscle function. So the fungal spore swims through the water with this flagella to the frog. It burrows into the skin and creates a structure called a sporangia. where it grows new fungal spores to be released into the wild. However, the damage created by the fungus on the skin prevents the frog from absorbing electrolytes, leading to electrolyte depletion. And because electrolytes are really important for muscle function and the heart is a muscle, electrolyte depletion can lead to cardiac arrest or heart failure. The disease has been so devastating to frogs worldwide, and chytrid has played a part in the decline of 500 amphibian species, or over 500 amphibian species. 90 species are confirmed or presumed extinct in the wild, and there's 124 species experiencing a 90+ reduction in numbers, so at least 90% reduction. This chart here shows the effects of the worldwide die-off, and it's from a recent publication by Shiel et al. In 2019. Each line of this chart is a species, and the color of the line tells us how much of that particular species declined. For example, these blue lines indicate those species that have declined about 20%. These yellow lines here indicate species whose numbers have declined 90%, and the red lines are species that are thought to be extinct. Now that I've given you all the gory details on chytridiomycosis, we can go on to our hunt for gold. Our hunt for gold starts in Panama, and which is this small country on this isthmus here that connects North America and South America. We work in partnership with the Smithsonian Tropical Research Institute in Panama City, which is Panama's capital. Now, the gold we're looking for is probably more rare than real gold. The variable golden frog is the national icon of Panama. Beloved by the country, there's an entire week dedicated to the celebration of the frog, including parades, a 10K race, and educational presentations. That's me in the parade because, if you get the opportunity to dress up like a frog in a parade, you don't think twice about that, right? However, the frog itself hasn't been seen in the wild for years. When chytrid spread through Panama, the species was already threatened by deforestation and the illegal pet trade, and then the emergence of the disease all but wiped the frog off the map. If you wanted to see them now, you'd have to visit one of the amphibian conservation centers that houses them. These frogs, along with many other species, are now in a captive breeding program where conservationists have bred several generations in hopes of one day releasing them back into the wild. But if the frogs are not able to protect themselves from the threats that still exist in the wild, the re-released frogs would most likely die. If we want to prepare these captive frogs for the wild, we need clues from surviving frogs in the wild. But as I mentioned before, it has been virtually impossible to find the golden frog in the wild. Besides searching for miles through the dense jungle, my lab has employed multiple strategies to try to find any trace of the golden frog, which include collecting stream water samples and filtering out environmental DNA. So environmental DNA are DNA molecules that can be found in the environment, like in the water. They're intact molecules that we can use, and if they're intact enough, we can find out where they came from. And I do this by using the thermal cycler that we talked about earlier. The thermal cycler amplifies the target DNA if it's present. So here I have, I'm filtering water that we collected from a stream. I'm using this hand pump here, and then a very fine filter that attracts and collects the DNA from the water. And then another tool we use to try to capture signs of the golden frog are these audio recorders. Last time we went to Panama for fieldwork, we took these recorders and strapped them to a tree. And they are programmed to record over a weekend. Then we sent the resulting audio file to the University of Pittsburgh, where our colleagues have invented a computer program that can pick out frog calls from a noisy recording. Since the golden frog has a very unique call, we should be able to detect its call if it is present in any of our study locations. One of the many places we use these tools is in the Omar Torrijos National Park in Panama. This national park used to be known for being a popular place to find the golden frog, and that's why they used to have the statue of the golden frog at one of the trailheads. The statue's still there, but it has since been painted green after the golden frog disappeared. At some angles, you can see some of the yellow paint peeking through. I had to snap this photo because it was a heartbreaking reminder of why we work there. So in order to find out how the golden frog could survive infection without chytrid, we need to be able to analyze multiple surviving frogs, like I said. So instead, we can use and investigate how other frog species have survived besides the golden frog. So as scientists, we need to perform the scientific method. So the scientific method, I'm sure everybody remembers from elementary school or middle school. Make an observation, and then you form a hypothesis, and then you create the experiment to test the hypothesis, and then you draw a conclusion. That's kind of a simplified view. So I'll show you how I, as a scientist, use the scientific method to find out how some frogs are surviving while chytrid is still present. So my lab has traveled to Panama for years to swab any and all frogs to see if they're infected with chytrid. From there, we can calculate the infection prevalence, which is the proportion of frogs that are infected with chytrid out of all the frogs we test. We follow a strict protocol to capture and swab frogs to see if they're sick. Tropical countries like Panama have only 2 seasons, unlike Reno, where we have 4 seasons. They have the wet, rainy season, and then they have the dry season. So with this species, we have observed that there is a seasonal change in infection prevalence between the 2 seasons, as shown in this graph, where the y-axis reflects infection prevalence. 1 means that 100% of them were infected, while 0 means that 0 of them were infected, and 0.5 means that 50% of them were infected. So, and then we have the 2 seasons on the bottom. So interestingly, about 3/4 of the frogs we swabbed over the course of 4 years now were infected in the wet season, compared to less than 50% in the dry season. So we wanted to find out if this might be because their immune system changes with the season too. So therefore, I've started, for my PhD research, I've started looking at some immune functions of frogs. So this is a diagram of a cross-section of frog skin. If you were to slice a frog in half and look at it under a microscope, you would see something similar to this, but maybe a bit less colorful. So we have here the epidermis, so this is the surface skin cells, and on top of the epidermis we have this mucus that frogs have, along with cutaneous bacteria. This is also where the BD zoospores will infect at the surface of the skin. And then underneath here you have the dermis. You also have some glands in the skin. And then you have the blood capillaries here, and then the organs that are a bit deeper. So the first immune functions I'm looking at are skin secretions, which are up here on the surface, and then also something called a complement, which is found in the blood there. So I hypothesize that these immune defenses might be helping the surviving frogs survive during their higher infection in the wet season. So the first immune function, skin secretions, you see how slimy this frog is? This is because frogs have 2 types of glands that release slimy secretions onto the surface of the skin. And skin secretions are found in the mucus of amphibians and contain toxins, bacteria, and protective proteins called antimicrobial peptides, which I'll just refer to as AMPs for now. Some AMPs have been found to slow or stop BD growth, which protects the animal from infection. Similarly, some AMPs have also been found to have antimicrobial activity with other pathogens, such as the influenza virus. This particular species here, the skin secretions will kill the virus in vitro in a petri dish. So that's pretty interesting. So same kind of concept. So my experiment for the skin secretions is to identify chemicals that from various species and compare them and see how the concentration changes between species and then also between wet and dry conditions. And then I'd also like to mix these skin secretions with chytrid spores to see if they die and how much of a killing power they have over those zoospores. So here is a little flow chart of what I would do to collect these skin secretions. So the frog is here in this dish, and he is releasing his skin secretions into the sterile water. Here are some peptides and some other molecules, and those molecules go in the mass spectrometer that we talked about earlier. And then I'll get this printout, which will give us some clues on what these chemicals are. And then additionally, we have some preliminary work that isn't by me, but is by a colleague. And he's kind of the, Doug Whittams is kind of the forefront of the AMPs and skin secretion research. So this is from a paper that he published in 2007. So you can, these kind of look confusing. I'll go over them. There's 4 different species of frogs here. And this is showing you how effective the skin secretions from each of these frogs can kill BD zoospores or chytrid zoospores. On the y-axis here, you have a number that is basically a proxy for bacterial growth. So the higher the number, the higher growth of bacteria, the lower number, the lower growth of bacteria. This square here represents the positive control. So that is a control we put into experiments to make sure that it worked correctly. We'll use a positive and a negative control. And these are basically known chemicals that we know will be positive and know will be negative. So as we can see on these 4 charts, they roughly tell us about the same thing, that the higher the concentration of skin secretions and antimicrobial peptides that there are mixed with these zoospores, the less zoospore growth. And I'm sorry, I actually said this was bacterial growth. This is actually fungal growth here, not bacteria. So the higher the concentration of AMPs mixed with these fungal spores, the lower the amount of fungal spore growth. So you can see the concentrations, which are down here on the x-axis. And as the concentration decreases, you see the zoospore growth increase. So that's basically just showing you there that, yes, these 4 species of frogs do have some skin secretions that can inhibit chytrid growth. So another immune response I'm looking at is something called complement, and I won't go over complement in too much detail. All you need to know is that complement is a collection of proteins that are found in the blood of animals, including humans. They help by killing bacteria, and they summon other immune cells to the area of damage or pathogen damage. To study these proteins, I took serum from bullfrogs and separated the serum from the blood. And that is where the complement proteins are located, in this yellow liquid here. So in order to study these proteins, I take blood from bullfrogs and separate the serum from the cells with a centrifuge. That's what the blood looks like before the centrifuge, and this is what it looks like after. All these blood cells in the centrifuge fly to the bottom of this, and then I take this liquid up here, and that's where the proteins are that I'm interested in. I use the serum to see how well the complement proteins can kill bacteria, which tells me how well the immune system is working. So in this case, I am actually using bacteria as opposed to zoospores, but I plan to use the zoospores in the future. So my experiment with complement involves a very similar experiment that was done with the skin secretions. So here I have a diagram that describes a serial dilution of complement of serum, of frog serum that we know has the complement proteins in them. So I have A has the highest concentration and H has the lowest concentration. So I would expect if this serum is inhibiting bacterial growth, there would be less bacterial growth here and more bacterial growth here. So the results of this experiment is pretty much exactly what I expected, luckily. So we have the experimental data points here. Again, this A through H are lined up with the A through H in the serial dilution, so you can really see how the dilution affected the growth of bacteria. So you have down here really low growth, up here the purple, that's the positive control. Way at the top, we know that that's all positive. There hasn't been any inhibitory effects on those bacteria. We have this negative control with dead bacteria. I heat kill them for 10 minutes in boiling water to make sure that they're not going to grow. And then I have the experimental here. And then I also have just the serum and the bacteria on the green. So as we can see in this graph, lower bacterial growth with the higher concentration of serum. So we see a similar bacterial inhibition with these molecules as well as the skin secretions, which is really interesting. So my future plans involve continuing to analyze these functions in frogs in the wild, as well as in the lab. Understanding how AMPs and complement activity affects BD infection dynamics will allow us to find out how frogs might be changing to fight off infection, and can also inform conservation and reintroduction programs in the future. Because many vulnerable species were taken into captive breeding programs, we need to know that if these species are reintroduced to their habitat, that they will survive, because BD is still out there and just as pathogenic as ever. Therefore, my future plans are to draw a conclusion. So I hope you've enjoyed my talk. I discussed my work and why I do it. I gave you some preliminary data to discuss what we already know, and then I took you along with us on a hunt for the golden frog. However, I did actually leave out something. So I was recently notified that a scientific paper I helped author was accepted for publication after peer review. Remember the audio recorders I mentioned earlier that we used? Well, they found the golden frog. Out of hours of recordings muffled by the sounds from the stream and other animals, our colleagues at the University of Pittsburgh, with their machine learning program, which is lovingly called the Repeat Interval-Based Bioacoustic Identification Tool, or RIBIT for short, was able to isolate and identify the chirp of the golden frog. And you guys will be the first ones to hear it. So here we go. Make sure you turn your volumes up. And that's the golden frog. You can hear the stream in the background, and then the unmistakable chirp. And that is basically analyzed and picked out due to a pulse repetition rate. So there's a pulse repetition in frog calls. It's actually a set of individual notes, but we hear it as a solid call. And that is what this machine uses and can understand and hear way better than the human ear can. So I'm really excited to show that to you guys, to share that with you. That's the first time that anyone has heard it. So you heard it here first. And with that, I'd like to thank Addison Whiteley, my little sister, who helped me with this presentation, and the Voiles and Baroto Labs that are so supportive of all the work I do. Thank you, and I'll take any questions.
Thank you very much, Keely. That's great. Super exciting to hear that frog noise at the end as well.
Awesome. I'm glad it was you guys could hear it well and everything. Yeah, it's my my text message notification tone now.
Great.
Yeah.
Now the world will hear the golden frog. That's wonderful. So we're going to open up to any audience questions. Feel free to type them in and we'll get them over to Keely. To kick us off, I wanted to ask, I know that you're still kind of in the middle of this research, but do you think that there will be some difference in the response to the disease based on frog species, that some species may be more resistant?
Exactly. And this is what makes the research a bit more complicated, because we're finding that each species has its own susceptibility. Even within the same genus, species can differ pretty significantly from each other, even though they're related. So it's almost like we have to find the answers for one species, but then we want to compare those to other species to really figure out how frogs as a whole. And I wouldn't be surprised if there was multiple immune functions that came into play depending on the species, and lots of other things that have yet to be discovered.
Mm-hmm. And based on their species, do different frogs have different amounts of secretions on their skin?
Exactly. Yep. So there's different chemicals based on what they do. So some of these skin secretions, like I said, are toxins. So like the poison arrow frogs, they have different toxins that are actually like pretty toxic. Whereas other frogs don't necessarily have toxins that can harm humans, and some of them do. But yeah, so we definitely expect there's a few known skin secretion molecules that we're aware of, but there's only a few species that have been tested. So there's definitely a lot of low-hanging fruits.
Yeah, lots of room for future research. So I'm wondering, I know that you're very interested in how a lot of different animals interact with their environments. How did this all lead you specifically to studying frogs?
Oh, well, it was mostly a interest in microbiology and disease dynamics in general. And I also grew up working in the animal care field. So before I started in undergrad, I was working at an aquarium and I was a curator there. So we were taking care of lots of fish. And then I learned all about the maintenance of those guys. And then after that, I went and worked with sea turtles for a season in Costa Rica. And it just kind of went along there. Those were all cold-blooded animals, similar immune systems, similar biology compared to like warm-blooded animals. And so I really wanted to find somewhere in grad school where I could work in the lab and also in the field and combine the hard microbiology work with the field work. And I just love it.
Great. And I'm wondering, for the students who are watching, if you can describe a little more what it looks like to be a lab scientist who also does field work. Do you have to go on location and collect samples, or is that shared within your lab? Obviously, you work in a place where we don't have golden frogs. So what does that look like?
Yeah, exactly. So with COVID, our trips to Panama have been canceled. Like the last time we went there, I think was December of 2019. So we usually go twice a year. We'll go in the winter, which is kind of approaching the dry season, and then in the summer, which is the wet season. And yeah, we haven't been able to get out there, but normally I would go to Panama and get samples and do all this work. We recently just got our last samples back that we collected from 2019. They mailed them to us finally. So we get to at least look at those samples because we did collect some skin secretions and lots of swabs. And yeah, so it's pretty much this particular thing is me and then my advisor, Jamie.
Wow. A big project for a little team. That's very interesting.
Yeah, I don't know if I might be biting off more than I can chew. Hopefully not. But
Right.
the point is in grad school to find something that you love. So
Yes.
even on those days when it gets really hard and difficult, you still have that passion there. So
Mm-hmm. And I wonder if you can talk about how scientists work together to create future solutions. So obviously you're figuring out which frogs may be more resistant to this, which may not be, and then where do we go from there to come up with solutions?
yeah, so we have lots of collaborators. Like as you saw with the audio recording, we're working with machine learning experts in universities across the country. And there's so many different ways that all of these experts can help us or are helping us in this race. So right now I'm kind of cross training in a chemistry lab to do the analytical side, and so I'm learning all of the stuff from that point of view. So I'm realizing how different chemistry deviates from biology when you work in the lab professionally. So I love all the cross training, and but I also love collaborating with others. There's a lot of people that have been working on it way longer than I have. So we also have a lot of collaborators in Panama, which include professors and researchers at the Smithsonian, as well as families that own ranches in the middle of nowhere where we might find in areas where we used to find the golden frog. So it's really a huge effort by every type of person in multiple countries. So it's quite exciting.
That is very, very exciting to kind of hear that all of these forces are coming together for this one little frog. It's pretty inspirational. So
Yeah.
we're gonna wrap up soon, Keely, but I wanted to leave us on an inspirational note for our students watching. If you are kind of interested in maybe going into studying amphibians or interested in biology but don't quite know where to go, what would you recommend for a student in that position?
Um, just not to get discouraged. It's easy in conservation to be overwhelmed and feel like, well, what can I possibly do? Like these problems seem so big. And I often question, what if my work doesn't come to fruition? Like, what if it doesn't work? And it's bound to not work sometimes. But then you have these little glimmers of hope, like this recording or different scientific findings that kind of give you hope and make you realize, yeah, this is actually doable. And you just have to commit to that and stick with other conservationists so they can keep you in good spirits as well, for sure.
Mm-hmm. Well, thank you very much for talking us through that, leaving some inspiration to our students, and we're very thankful and grateful that you work in conservation. So thanks for being here to tell us more about what you do.
Thank you so much. It's been great to talk to you guys.
Great. Well, I hope everyone has a great day, and I'll talk to you soon.
Awesome.